Hello, everyone, and a very warm welcome to this year's PacBio workshop as part of the PAG conference. My name is Jonas Korlach, and it is a great pleasure and privilege to once again host and moderate today's event. We have an exciting program for you today. My colleague Jake Brandenburg will tell you in detail about all the latest products and applications that we offer from PacBio, and then you will hear from 5 researchers sharing their experience applying PacBio HiFi sequencing to a wide spectrum of different applications in the plant-animal genome sciences. Before I hand the stage to the speakers, please allow me to make a few introductory remarks. You have all been very busy in 2024. This graph shows the accumulation of raw PacBio sequencing data for the last 4 years or so, and you can see that there's a very steep increase. About 2 months ago, we celebrated a milestone of over 100 petabases. That's 100 million billion bases that were generated with the PacBio technology. For those of you among here who have contributed to this graph, thank you very much. We really appreciate your business, and we feel privileged that you have chosen PacBio technology and the employees of our company as a partner in your research. One other thing I want to say about this graph is that 45%, so nearly half of all the PacBio data that have ever been collected were done just in the last year in 2024. This has resulted in a large number of peer-reviewed publications. You can see the curve has a very similar shape. It has no y-axis because it has become increasingly hard to get the exact number. As PacBio has become more mainstream, the fact that PacBio sequencing was used now is in the supplement rather than in the title or in the abstract. But we are there's a conservative estimate that there are now over 10,000 publications. With regard to the plant-animal genomics sciences now, we have seen contributions across the globe and in all the different areas of plant and animal genomics, so agrogenomics, biodiversity, and environmental health. Starting with agrogenomics, by now, essentially every crop and livestock species has received a high-quality PacBio HiFi-based reference genome, and we recognize this fact at our booth. If you haven't had a chance to go by, please check it out. In that, on the left side, there's a few examples of fruits and vegetables and animal products that have received a PacBio HiFi genome reference, and a few trivia and so forth. I do want to mention, since I got some questions about this, all the food, all the edibles will be donated after the conference to the San Diego Food Bank, which I understand right now is transferring most of their donations to the Los Angeles Food Bank to help with the victims of the terrible wildfires there. Increasingly, though, one reference genome for a species is not enough. So for the things that we eat, we've seen the movement towards pan-genomes. And so this was justified the cover of Science in the summer of last year. And if you've been to any of the Nightshade workshops on Saturday and on Sunday, you saw Zach Lipman and his colleagues and Mike Schatz present on higher quality pan-genomes. Mike told me that the preprint that you see on the right is now accepted for publication and should appear in a few weeks' time, really exemplifying a new era in crop pan-genomes where you can now really see the paralogs that are so crucial for the foundation for crop engineering. And in the context of this workshop, Rebecca and Shannon will talk about aspects of this part of agrogenomics and will represent examples of how they use PacBio sequencing to improve crop sciences in their research. Of course, then we also want to have a good understanding of the things that eat the things that we eat. And so this is a major focus in Scott Geip's research from the USDA in terms of studying the pests that can cause billions of dollars in damage. And then finally, we need to sequence the things that eat the things that eat the things we eat. So that's a long-winded way of saying that's biological pest control. And now you're certainly more deeply into the tree of life and biodiversity. Shannon and Mark are going to talk about that. But then, of course, beyond that, also about biodiversity in terms of characterizing biodiversity as a foundation to conserve biodiversity. So the two themes for us at the workshop are sequence to eat well at scale, and sequence to conserve. And so Shannon will talk about oceanomics, and then Mark Blackson will talk about Tree of Life and other associated programs to really bring HiFi sequencing to a scale where it's in production setting for hundreds and thousands of species that now get a high-quality genome. Increasingly, of course, it's been recognized that the environment is critical for plant-animal health. And there was a really nice paper, a review paper that just published last month on describing all the different ways that HiFi sequencing has unlocked the potential of metagenomics in these different areas that are depicted here schematically on the left. Of course, that will be the focus of Holly Gantz's talk in terms of characterizing animal biomes and gut biomes. And so she'll talk about applying the PacBio technology for a commercial kit that you can buy or can be ordered by the veterinarian to better understand what might be the case with your domesticated animals. Scott will also have a story about full-length 16S sequencing to better understand microbiomes. And then we'll have a dedicated talk by my colleague Jeremy Wilkinson tomorrow at 11:10 if you're interested in this area using the Kinnex 16S full-length kit. Please check out his talk as well. We are seeing new and improved solutions developed by the community, either by themselves or in partnerships, or we are developing them. And I just want to highlight 3 examples here. Best-in-class genome annotation. This is a paper that just published 2 weeks ago in Genome Research by Anna Canessa and members of the LR-GASP consortium with the latest example of a technology benchmark comparison, comparing all the different technologies, Illumina, ONT, and PacBio, and finding that the best performance was achieved when using the transcriptome inferred from PacBio data as experimental evidence, and finding that incorporating PacBio transcripts significantly outperformed traditional methods for genome annotation. In the paper, they applied this to the Florida manatee and uncovering thousands of new genes and isoforms that were not known before. Reducing the input requirement. My colleague Jake Brandenburg will talk about the blog that we posted this morning about an improvement to our, what we call ultra-low input protocol that now goes down to 1 nanogram of DNA input that is now compatible with HiFi sequencing. And then Mark will describe pushing that boundary even further below 1 nanogram with the picogram input multimodal sequencing method, or PIMS method. And if you allow me just a personal anecdote, this little guy right here was the very first critter that was subjected to what would later become the PIMS molecule. This is a tiny little flatworm, smaller than a millimeter. And Chris Lohmer, who was at Sanger about 4 years ago, and he, of course, Chris Lohmer is the developer of the PIMS method, contacted me and asked, can we try PacBio sequencing on this myofauna, these very, very tiny species. And so we tried it, and it worked really well right off the bat with this first one. And so it'll be really exciting to hear from Mark now how this method has now been applied to many, many more specimens and really breaking into a domain of the tree of life that's been really difficult to assess at high quality. And then Scott Geip will tell you about sci-fi sequencing. So this is Hi-C with HiFi data, showing that it is now possible to use one technology. I should say that this is a method that was developed by Megan Dennis at UC Davis. And then Scott collaborated with Megan to apply her method to single insect specimens. So Scott will tell us that it's now possible with one technology, just PacBio, you generate HiFi data, you generate sci-fi data for chromosome scale, high quality assemblies that are haplotype resolved. So you see there, you get 2 copies of each chromosome for diploid organisms, of course. Our commitment to all of these efforts is to give you ever increasing capabilities. And so in this workshop, we're going to talk about how we further scaled and expanded the sample types with the new chemistry, with the Spark chemistry on Revio, to provide greater accessibility to individual researchers and to laboratories with the new benchtop Vega system, and enhancements to all aspects of sequencing projects for either system, for both of these platforms, and that are in the area of extraction, automation, bioinformatics, and reducing the input amount. And so this is actually the outline that my colleague Jake Brandenburg will use to now come up on the stage and talk about all of these three aspects in a little bit more detail. So, Jake?